one of the most recent technologies to come out into the market, with animal cloning many different possibilities have been opened up. Today, we will explore the history of an animal cloning itself, along with other animals that have been cloned.
The animal world can be considered as a wondrous world. With the advancement of science and technology, animal cloning has been made possible. A few decades ago, people were always talking about agricultural cloning, which is the cloning of plants. However, it has been taken to a whole new level. A whole animal can be cloned. A whole live animal, think about that. What is animal cloning? It is the creation of a new multicellular organism genetically identical to another. In essence, this form of cloning is an asexual method of reproduction, where fertilization does not take place. Asexual reproduction is a naturally occurring phenomenon in many species, including some insects and animals. Scientists have made some major achievements with cloning, including the asexual reproduction of sheep and cows. However, there is a lot of ethical debate over whether or not cloning should be used. Reproductive cloning generally uses somatic cell nuclear transfer, otherwise known as SCNT, to create animals that are genetically identical. This process entails the transfer of a nucleus from a donor adult cell to an egg from which the nucleus has been removed, or to a cell from a blastocyst from which the nucleus has been removed. If the egg begins to divide normally, it is transferred into the uterus of the surrogate mother. Such clones are not strictly identical, since the somatic cells may contain mutations in their nuclear DNA. Additionally, the mitochondria in the cytoplasm also contains DNA, and during SCNT, this mitochondrial DNA is wholly from the cytoplasmic donor's egg. Thus, the mitochondrial genome is not the same as that of the nucleus donor cell from which it was produced. This may have important implications for cross-species nuclear transfer, in which nuclear mitochondrial incompatibilities may lead to death. Artificial embryo splitting, or embryo twinning, a technique that creates monozygotic twins from a single embryo, is not considered in the same fashion as other methods of cloning. During the procedure, a donor embryo is split into two distinct embryos that can be transferred via embryo transfer. It is optimally performed at the 6 to 8 cell stage, where it can be used as an expansion of IVF to increase the number of available embryos. If both embryos are successful, it gives rise to monozygotic twins.
Now, here is an example of a cloned domestic animal. Dolly, a fin dorsal eel, was the first mammal to have been successfully cloned by an adult cell. Dolly was formed by taking a cell from the udder of her biological mother. Her biological mother was 6 years old when the cells were taken from her udder. Dolly's embryo was created by taking the cell and inserting it into a sheep's ovule. It took 434 attempts before an embryo was successful. The embryo was then placed inside a female sheep that went through a normal pregnancy. She was cloned at the Roslin Institute in Scotland and lived there from her birth in 1996 till her death in 2003 when she was 6. She was born on July 5, 1996, but not announced to the world until February 22, 1997. And today, her stuffed remains were placed in Edinburgh's Royal Museum, part of the National Museum of Scotland. Dolly was publicly significant because the effort showed that genetic material from a specific adult cell, programmed to express only a distinct subset of its genes, can be reprogrammed to grow an entirely new organism. Before this demonstration, it had been shown by John Gurdon that nuclei from differentiated cells could give rise to an entire organism after transplantation into an enucleated egg. However, This concept was not yet demonstrated in a mammalian system. Cloning Dolly the sheep had a low success rate per fertilized egg. She was born after 277 eggs were used to create 29 embryos, which only produced 3 lambs at birth. Only one breech later. 70 cows have been created and 1/3 of them died young. Prometheus took 814 attempts, notably, though the first clones were flopped. No adult clone frog has yet been produced from a somatic adult nucleus donor cell. There were early claims that Dolly the sheep had pathology resembling accelerated aging. Scientists speculated that Dolly's death in 2003 was related to the shortening of telomeres, DNA protein complexes that protect the end of linear chromosomes. However, other researchers, including Ian Wilmot, who led the team that successfully cloned Dolly, argue that Dolly's early death due to respiratory infection was unrelated to deficiencies with the cloning process.
The reconstruction of functional DNA from extinct species has been a dream of some scientists. Currently, cloning techniques have an average success of less than 5% even with familiar species. These are difficulties faced and is a very slow process to save species by cloning. In 2001, a cow named Bessie gave birth to a cloned Asian cow, an endangered species, but the calf died after 2 days. In 2003, a banteng was successfully cloned, followed by 3 African wildcats from a thawed frozen embryo. These successes provided hope that similar techniques using surrogate mothers of another species might be used to clone extinct species. Anticipating this possibility, tissue samples from the last Ricardo Pyrenean ibex were frozen in liquid nitrogen immediately after it died in 2000. Researchers are also considering cloning endangered species such as the giant panda and cheetah. In 2002, geneticists at the Australian Museum announced that they had replicated DNA of the thylacine Tasmanian tiger, at the time extinct for about 65 years, using polymerase chain reaction. However, on February 15, 2005, the museum announced that it was stopping the project after tests showed that the specimen's DNA had been too badly degraded by the ethanol preservative. On 15 May 2005, it was announced that the thylacine project would be revived, with new participations from researchers in New South Wales and Victoria.
In January 2009, for the first time, an extinct animal, the Pyrenean ibex mentioned above, was cloned at the Centre of Food Technology and Research of Aragon, using the preserved DNA of the skin samples from 2001 and domestic goat egg cells. The ibex died shortly after birth due to physical defects in its lungs. One of the most anticipated targets for cloning was once the woolly mammoth, but attempts to extract DNA from frozen mammals have been unsuccessful. Though a joint Russo-Japanese team is currently working toward this goal, in January 2011, it was reported by Yomiuri Shimbun that a team of scientists headed by Akira Iritani of Kyoto University had built upon research by Dr. Wakayama, saying that they will extract DNA from a mammoth carcass that has been preserved in a Russian laboratory and insert it into egg cells of an African elephant in hopes of producing a mammoth embryo. The researchers said they hope to produce a baby mammoth within 6 years. Scientists at the University of Newcastle and University of New South Wales announced in March 2013 that the very recently extinct gastric brooding frog would be the subject of a cloning attempt to resurrect the species.
It is currently an emerging market and it is slowly becoming more popular in the biotechnological field. However, cloning should be done with precaution as the results usually will be unexpected.